as we were discussing about it, that these proteins are there, and this causes, that is, the scaffold assembly is there, factors are there, and they are going for the pre-replicative complex. So this is the next stage which will be there. This pre-replicative complex assembly during G1 stage of the cell cycle is required prior for to the activation of DNA replicating during S phase, because S is a synthesis phase. Okay, so during synthesis phase, whatsoever has to be done, before that all the preparation has to be ready, and the removal of the last part of the complex, that is ORC1 from the chromosome at the metaphase, is the part of regulation of mammalian ORC. That is how this is happening, to ensure that the pre-replicative complex, that is original recognition complex, is there, that is regulated, to ensure that pre-replicative complex formation prior to the completion of metaphase is eliminated. So this complex is again destroyed. This complex is again, before metaphase it is over, so that if the next cell has to be generated, again it starts from the scratch. I will not discuss the details about each and every protein what they are doing. Okay, these are various protein, and we have seen that they are important. They are part of the ORC, that is origin recognition complex, and it is an essential step. So we will not go in more detail, but one thing, very important thing has to be kept in mind in our always, that see, when we say that proteins are playing very, very important role, so we should understand how the proteins are basic part of our body. Though we say that, or though we have studied everything in biochemistry, that we have got lipids, we have got carbohydrates, definitely each and everything is playing role. But again, very, very important thing is, if lipid is there, lipid has to be metabolized by enzymes, and all enzymes are protein. If it is carbohydrate, yes, carbohydrates are very, very important. They are essential for our energy production. Without energy, the organism will die. But again, whatsoever things are there to convert this carbohydrate into energy, you need enzymes, and for all enzymes, again, these are the protein. So if we have to make that, we must understand that proteins are playing very, very important role. So they are responsible for their own formation also, because these proteins are responsible for all these enzymes, whether it is DNA dependent DNA polymerases or DNA dependent RNA polymerases, which are responsible for DNA synthesis or replication, or those which are responsible for RNA synthesis or transcription, what we call. In all cases, proteins are playing important role. Now, why we are stressing this again and again? The reason is this, that anything goes wrong in this, if any structure goes wrong in any kind of protein, then that will cause alteration of the activity. And if this alteration is there in activity, that is bringing out pathology. Okay. So therefore, these proteins are very, very important. All these proteins are there, and it is a cycle we can have. That DNAs basically are there, or they are part of chromosomes or genetic material. These DNAs are responsible for coding for the various activity which includes protein, and because of that, these all proteins are playing important role, again, as we are seeing here, in the DNA replication or RNA formation. So these are very, very cyclic process, interdependent process, and therefore we should remember this, that they are very important one. So these are the CD6 protein, CDT proteins. They are playing important role in all ORCs. They are playing role, and they are particularly making important complexes like ORC, CD6, CDT1, and MCM. So these complexes, these are the microsome maintenance protein complex. These all are important one, and how much important they are, we can realize from that, if we'll see that separate articles are there for each, which that much detail we don't want to go for. If we go for compiling each and every article in that sequence, it can be very, very big book from Wikipedia itself. which we don't want to go for this much details. So these are various proteins which they are participating, and then the next thing starts to that is initiation complex. First one was ORC, and then the next one is there, that is ORC. So what happens before that, just have a glimpse of it. That is this MCM are there, that is microsome maintenance protein there, MC, yes. So MMP proteins are there, I'm sorry. So this MMP protein, or also called as a MCM, mini chromosome maintenance, that is MCM 227, which we have seen. So these MCM proteins, they are playing important role, and these are they are making the nuclear localization of the these proteins. In yeast cell it has been found. They are present in the nucleus, particularly during G1 stage and S phase of the cell cycle. That means they have important role to play during G1 stage and S stage, and they are exported to the cytoplasm during G2 phase and the M phase of the cell cycle. A complete and intact 6 subunit MCM, that is MCM 2 to 7 we have seen. These are the 6 subunit, MCM 2, MCM 3, MCM 4, 5, 6, 7. So like that 6 subunit is required to enter into cell nucleus. Okay. So they are very, very important, and as we have studied earlier that this yeast plays, or this yeast has helped to learn a lot about this process. So in that yeast, what happens with the CDK, it has been mentioned, which we will not discuss now. We will go for only human being. This is just an experimental part which has helped to understand the basic process well. So next is the initiation complex. This initiation complex will have another set of proteins. So during G1 stage of the cell cycle, replication initiation factors, origin, ORCs, CD6, CDT, and MCM 2 to 7. They will bind sequentially to DNA. They will form the pre-replication complex, which we have seen earlier. And at the transition of G1 phase to the S phase, next to G1 is a S phase. The S phase specific CDKs are there, that is cyclin dependent protein kinases are there, and DDKs are there. They will be becoming activated. They will do the job. They will transform this pre-RC, that is complex, that is pre-replication complex, into an active replication fork. Fork means कांटे का चमचा आता है जिसको दांत दिखते हैं ना, उसको भी fork कहते हैं. So this is the fork, replication fork, or comb-like structure. So this replication fork is depending on these two important, that is cyclin dependent protein kinase and DDK, that is CDC7 DB4 kinase, which are there. So these important kinases will transport this pre-RC into an active replication form. During this transformation, pre-RC is disassembled with the loss of CD6, creating the initiation complex. So thus initiation complex is formed after loss of CDC6, and in addition to the binding of this MCM proteins, cell division cycle CDC45 protein is also essential for initiation of DNA replication. It has been shown that MCM is critical for loading of CDC45 protein onto chromatin, and this complex containing both MCM and CDC14 is formed as the onset of S phase. So thus there is a transformation from, because of this, all these proteins CDC14 and the pre-RC complex, that is this replication complex is there. So these are how they are making it into the next stage, that is G1 to the S phase, that is synthesis phase. I will not, as I told you, will not go into detail about these proteins. There are variety of proteins are there. We have seen MCM 2 to 7. This is now MCM 10, which is participating, and these are important kinases. We shall discuss these kinases when we shall discussing about various pathways, but they are important one. They are playing very, very important role, particularly at the onset of S phase. We will have just glimpse of this kinases, because as I told you, they are important for many things, and we can have many important intervention with these CDKs or kinases. Okay, so therefore we'll have some more attention on these kinases. So at the onset of S phase, we have seen that this PRC complex, that is pre-replicative complex, has to be activated, and these are two S specific kinases are there, which we have seen earlier, that is DDKs and CDKs. These are doing a job, that is they are the catalytic subunits, and they are doing the catalytic activities, particularly in eukaryotes. They are required for the onset of S phase of the cell cycle. They are required for the loading of CDC45 onto the chromatin origins of the replication, and CDC45 plays a very important role in the next stages, S phase onwards. The target for binding DDK kinase is the MCM complex, particularly MCM2. We have seen MCM2 to 7 are there. These are the 6 MCM proteins are there. While the DDK targets the MCM complex, and its phosphorylation leads to the possible activation of the MCM helicase activity. While the CDK is acting on various things like CDC45, already we have seen, and it is the target of the CDK action, and because of that this causes CDK dependent phosphorylation of CDC6, and thus it causes, whenever phosphorylation is there, that means it is activation, and thus this activation of CDC6 will cause the entry into S phase. So thus CDK dependent and DDK dependent kinases, they are very very important, or CDK rather, that is cyclin dependent kinases, and this DDK, that is this particularly kinases, that is DD protein are there. So these kinases are important. So these two kinases, as I told you, they are holding a key in the next stages. Again, there are many other proteins. I will not go into more details, and I am not even expecting that you should go for that. So this was the first stage is over. We have entered into the synthesis phase, and this synthesis phase will cause elongation of the size to what we call it is now replisome. That is, this replisome will cause now, because it has initiated now one after another molecules will be added into that, this one after another these nucleotides will be added into. We know that these are the nucleotides which are added, ATGCs. So this ATGC will be added one after another, and see how many proteins are playing role. This is again another figure which shows the next, this one. So one after another this leading strand is there, and there are variety of enzymes, proteins, CDC45 we have seen these MCMs we have seen, these are the 6 proteins we have seen. Okay, so these all are making this activity, and because of that this leading strand is making the lagging strands. These are the RNA primers are there, primases are there. All of these are making one after another activity, and thus it is leading for the elongation of the cycle. So this repliosome complex, and there are several associated proteins are there which are responsible for the elongation. So this causes elongation, and thus the cell goes into this is a synthesis phase, elongation is. So thus the cell now is in the S phase of the cell cycle. This elongation complex is formed, and the cells, as I told you, they are passed into S phase, then becomes a replisome, and this eukaryotic repliosome complex is responsible for coordinating DNA replication. Then replication on the leading and the lagging strand is performed by DNA polymerases epsilon, DNA polymerases delta, and many other factors are there, that is claspin, AND1, replication factor C, and many other things are there. We have seen so many factors, so many things are there. We will not go into more detail. We should just have that there are so many factors are there. Only important proteins and important enzymes, that is CDK or DDK, we should be keeping in mind. I am not expecting, because we don't want to be geneticist or expert in the genetics, and we don't have that much time also. We have got very important and other topics also, but since they are important, or this role is because future is now going towards this, and therefore it's better we should be having at least some idea about it. So what has happened in that, these replisome factors are there, as I mentioned this. They will be causing a regulation of the polymerases functions. There are various polymerases, we call them DNA dependent DNA polymerases, and we have seen their name, that is epsilon, delta, alpha. There are many such. So they will be doing this job. There is a coordination of DNA synthesis with this and unwinding of the template strand by this complex, which we have seen as in the pre-replication complex or pre-RC. We have seen the role of all these important proteins, CDC45, MCM, this GINS we have not seen, but they all are making complex, and the DNA is unwound, the twist number decreases. So there are many twists. It is a very, very twisted and complex, rotated around histones in nucleosomes. So it is unwound, just like it was. Histone is acting as a reel, suppose. So one reel thread, another reel thread or bead, like that. So this is unwound. So this number of twists are decreasing, because whenever there is a twist, you can understand, it's very simple. If the twist is there, suppose you have got a thread or a rope like structure, and in that we have got the thread is made of again two smaller fibers which are twisted together. So if they are twisted together, and if you give the twist to the thread, you will not able to separate them. Simply you just take a thread whenever it is available, and you will try to separate the strands of that thread. Okay, it is twisted, and therefore it is appearing as a single thread like structure, but it is already made up of strands. Now if you try to separate strand, and if the thread is a straightened one, you will find that easily you can do it. But if it is not straightened one, if it is already woven on the your reel of the thread, then you will find that it is not that easy. Okay, so that means that then it will not able to unwound. And if the strands are not separated, then the next complementary strand cannot be synthesized. So all these functions are very, very important. The coordinating DNA synthesis with the unwinding of the template strand by these proteins. When it is unwound, the twist number decreases, and to compensate this, the right number that decreases. What is this right number is? The several competing notions are there about right, or it is also called as WR. So it is purely property of oriental link diagram, and it has got this kind of mathematical knot structure is there. So right number is a mathematical number is there, which speculates or which gives some idea about the how many coils are there, or how many super coils are there after coil. Again, one thing you can try about, you just do one thing is, you have got a thread, you put the you take the part of a thread, you take two parts and try to coil it, try to twist it. If you twist it too much, then you will find that it get coiled automatically. Okay, so one coil after another coil after another coil, that becomes more and more coils, means first coil, then coil is coiled again, these two are having one coil. So this coil after coil, they are called as super coils, and these super coils is responsible, or they are important, because this is a reason behind their packaging or their compression. So these are there, these super coils are there. The super coil will cause DNA replication halting, or because this, if the super coils are there, they may form the knot, and if they are not removed, then these super coils will not allow the DNA replication. So topoisomerases are very, very important. They will responsible for removing these super coils ahead of the replication. Just for the idea, I will repeat, what you try to do is, you try to separate thread strands, and you see all this action while reading all this. So you will understand what is super coil, you will understand, only thing is, you will not have enzymes over there which are doing it. But this is how these things are important and acting over there. So this replication, these replisomes, they are responsible for copying the entire genomic DNA in each proliferative cells. The base pairing and the chain formation reactions are there, which will form the daughter helix. They are catalyzed by the DNA polymerases. As we have seen, they are very, very important. These enzymes will move along the single-stranded DNA, and they allow for the extension of the nascent DNA strand by reading the template strand. So one strand is providing as a template, and the complementary will be there. Just like if there is a A, then the G will be formed. If there is a T, then A will be formed. Okay. So like that, if there is a G, then there will be C will be formed. There is a C, then G will be formed. So like that, as per this ATGC we have seen. So this complementary, on that basis, on the basis of the nucleotide, the complementary strands will be formed, and it will be reading the template strand. It will allow the incorporation of the proper purine or pyrimidine, whether it is adenine, guanine or thymine, cytosine will be there. They will be added. They will be activated free deoxyribonucleotides will be there, which will be there in the cell, and they will be present as a DTPs, DNTPs, that is deoxyribonucleotide triphosphates are there. These free nucleotides are added to the exposed 3-hydroxyl group on the last incorporated nucleotides. So nucleotide is added and the sugar is added into that. The pyrophosphate is released from the DNTP, that is deoxyribonucleotide triphosphates is there. From that it is there. It will generate the energy for the polymerization. So they have self-sufficient energy for that. So they do have their enzyme system, which is using different than ATPs. Instead of ATPs, they are using nucleotide, means not only adenine but other nucleotide triphosphates are also there, which are able to release phosphate, and because of that, or just like ATP to ADP, we there and we get energy. So here deoxyribonucleotide. So nucleotide means any nucleotide for that. So from that, this energy is generated, and this energy is making or giving the energy for polymerization reaction, and this is also exposing the 5-monophosphate position, wherein this covalently bonded to the 3-oxygen, and thus the linking is going on. So incorrectly inserted nucleotides can be removed and replaced by correct nucleotide in an energetically favorable reaction, because again energetically favorable means we have to see that if you will see how the AT and GC, or how means adenine, thymine or guanine, cytosine, they are making complex. They have their own set of bonding is there, and because of their the position, or because of their chemical structure, adenine cannot form with the cytosine. Okay, only guanine can form. So similarly, this is energetically or accordingly, which is able to make the proper bonds. So this property is vital for the proper proofreading. This is again, proofreading is very, very important thing. What is proofreading? If anybody knows, proofreading is what? Suppose somebody is writing book, or somebody is writing an article, somebody is writing something. So in that, maybe because of all this process, there are some grammatical or typographical errors can be there. So there is some person who is doing this proofreading, and he is finding out that no, there is a mistake, this is a mistake, this is a mistake, here the grammar is not correct, here the words are not properly used, here the maybe capitalization is not proper. So this thing is there, that is called as a proofreading. Okay, if proofreading is not there, then it can make the change in the sense of the word, because basically everything is done for the communication, isn't it? Suppose my pronunciation is not correct, or if I am writing something, there is a very famous joke I remember about is that in previous days there were telegrams, and most time it was made in a short one. So it was somebody made telegram that Chacha ji Ajmer gaye hain, because some inquiries that at that time telephones were even not less, they were very less. At very few people telephones were there. So telegram were there, that is go to the post office and write in typical words. We are fortunate, at least we have seen this. Your generation might not have seen this. So that is telegram was there, and people were trying to write it as short as possible, because every words were charged for that. And then once it is given to this post office, this will put to the teleprinters were there, just like messages now you are putting. That teleprinter will print it at that other place, and then from nearest post office it will be delivered to your house. That was called as a telegram. So if it is proofreading is not done, if it is not done, or if there is a typographic mistake, I remember that there was a joke there, that is somebody tried to send that Chacha ji Ajmer gaye hain, and the while printing or maybe while writing it becomes Chacha ji aaj mar gaye hain. So only there is a difference in the Ajmer and Ajmer. So that if it is not seen, what will that made a big chaos in the other place. Okay, and the person has tried to inform that he wanted to go home, but he had some important work at Ajmer. So before going back to home, he will go to Ajmer, and that he tried to inform, because that time phones were not there. So this can happen. This is what proofreading is there. Similarly, we know we have to convey the message. What is the message we are conveying? Because DNA, RNA are responsible. DNA is responsible for making RNA. RNA are responsible for making protein. Proteins are responsible for variety of body functions. And if this proper message is not going, okay, then what will happen? Just like Chacha Ajmer gaye, hai na? So the message was sent so that they should be having a peace of mind, that don't worry, he will come after 3 days because he had to go to Ajmer. Instead of that, the message received that the person has died, and everybody started crying like that. So this is how the proofreading or the sentence making is important. If you that is what basically what message you are trying to give, it will be making. So the property is there that will make the proofreading is there, then repair is there, and that will be important for that. That will be important for during DNA replication. So this is very, very important, and the next topic is replication of fork, which we shall be seeing later. I hope this much you have understood. This is important one, and if you have any difficulties, as I told you, we are not going into more and more depth. We are just trying to get the at least whatsoever is superficial information about the drug. So please go through this. If you have any difficulties,